all participants are present here. Should we start? So this tutorial is about the identification of non-coding RNAs, interaction of long non-coding RNAs with genes and other non-coding RNAs. Okay. So according to you, what are the major non-coding RNAs and why are they called non-coding in any species? Only introns? Introns, promoters? Okay. Okay. Non-coding RNAs are derived from which regions of the genome? Can anybody tell about it? Non-coding RNAs are transcribed from which region of the genome? Wow. Nobody know about it? Non-coding RNAs are derived from any region of the genome. Exon, intron, promoter, enhancer, any region. There are some specific properties that they are called non-coding. First is the common, that is they do not code any functional protein. They can encode small peptides but not functional proteins. Because in some recent studies it has been published that non-coding RNA can also encode small peptides with a small function. But a fully fledged functional protein cannot be formed by any non-coding RNA. So what are the major types of non-coding RNA that are present in the organism? एक तो हमने आज ही किया है, माइक्रो आरएनए, और कौन कौन से होते हैं? लॉन्ग नॉन कोडिंग आरएनए, एसआई आरएनए, राइबोसोमल आरएनए, एसएनआरएनए, circular RNA. Recently, circular RNA, the class of non-coding, that is circular RNA, has also been discovered. So these are the major non-coding RNAs which are present in any organism. So these are some basic points but very important points about non-coding RNA. functional proteins are encoded by only 2% of total transcripts, while remaining transcripts are considered as noise because of their non-coding ability or poor conservation. Previously, the only research is based on coding part, that is 2% of total transcripts. But now the research has widely occurred in non-coding part also. However, these non-coding RNAs form major branch of regulatory network of gene expression. Like microRNA, other non-coding RNA do not have any direct function. They also do their function via some gene. Like microRNA, it will target the gene, and through gene it will do its function. Likewise, other RNAs also, they also do their function by affecting some gene expression. Depending upon their function, non-coding RNAs are classified as housekeeping non-coding RNA and regulatory non-coding RNA, of which ribosomal RNA, transfer RNA, small nuclear RNA, small nucleolar RNA come under housekeeping or constitutive RNA. जो ये चारों RNA है, they come under constitutive. Constitutive means always working, always giving the expression or always transcribing. In addition, regulatory RNAs are further classified depending upon their length into short RNA such as siRNA, small interfering RNA, microRNA, PV interacting RNA, piRNAs, and long non-coding RNA. So these three, siRNA, microRNA and piRNA, these are short regulatory non-coding RNAs, while long non-coding RNAs are having longer length as compared to these. Then a newly identified circular RNAs are also included into this family recently. Okay? So this is the basic of non-coding family of RNA. Is this clear? Any doubts? So these are the non-coding RNAs and they are further classified according to their function. Now in recent years, advancements in high throughput sequencing and bioinformatic tools lead to the identification of various regulatory non-coding RNAs such as microRNA, long non-coding and circular RNA. Major regulatory RNAs are 3 RNA: micro, long non-coding and circular RNA. Circular RNA can also sometimes called as circular long non-coding RNA because they are longer as compared to the short RNAs. These non-coding RNAs emerged as direct or indirect regulators, majorly indirect regulators of gene expression through chromatin remodeling, transcriptional and post-transcriptional regulation. MicroRNA target various defense-related genes, and now I have also covered it previously that microRNA target the genes and affect its expression, but now microRNA can also target lncRNAs, long non-coding RNAs. They can bind to the, they can show complementarity to the long non-coding RNA sequence and they bind to it and cleave. So microRNA can also regulate the mechanism of the plant, defense mechanism of the plant or any mechanism of the plant by also affecting the lncRNA, because that lncRNA can also regulate some genes and microRNA is regulating lncRNA. So automatically the expression of these genes were also affected. Is this clear or not? MicroRNA target various defense-related genes and lncRNAs. Interestingly, both lncRNA and circular RNA can also act as sponges for microRNA and inhibit their function. This is very new. Both the lncRNA and circular RNA, basically research is now taken up pace on these lncRNA, circular RNA, and it has been discovered that lncRNA and the circular RNA, like microRNA, is binding the target. Now what they do, they can act as a sponge to the microRNA, means either lncRNA or circular RNA, they show complementarity to the microRNA, and then they go and bind to microRNA. So that microRNA cannot be able to bind to its gene or cleave its gene. So this is the meaning of this line, that both lncRNA and circular RNA can also act as sponges for microRNA. Means they absorb like a sponge. They just show the complementarity to the microRNA and they go and bind to microRNA. Now microRNA is busy with lncRNA or circular RNA. Now it cannot be able to target the gene. Clear or not? Sorry. But long non-coding RNA is affecting some other gene in another way. But the gene that is targeted by microRNA is now not anymore targeted, because microRNA is busy with long non-coding RNA. If I talk about the genes that are showing the, if I talk about lncRNA, that lncRNA is affecting the expression of these genes, then it will be affected. But I am talking about the gene that is targeted by microRNA. That will not be targeted because microRNA is busy. Now it can be occur that the expression of that gene is inhibited at some level, at very minimal level, because it can occur that some microRNA molecules can reach that target gene. But the expression, the inhibition of expression is very less as compared to the other pathway. Is this clear or not? जो lncRNA और सर्कुलर RNA है वो माइक्रो RNA के रास्ते में आ जाता है, एंड फाइनली वो जो माइक्रो RNA है वो अपना जो उसका स्पेसिफिक टारगेट था उसको क्लीव नहीं कर पाता. दिस इज द मीनिंग ऑफ दिस लाइन. मोर ओवर, अब now there is one more function of lncRNA. These are very interesting molecules. lncRNA can also act as precursors of various microRNA, because lncRNA, they have more than 200 nucleotides, and these nucleotides are very active. They can also act as precursors of microRNA. They can also act as sponges to microRNA. They can also be targeted by microRNA. So these are three interactions of lncRNA with microRNA. So these lncRNAs can also act as precursors of various microRNA. In brief, non-coding RNA make complex and intermingled networks that can be exploited to enhance disease resistance in plants. Disease resistance, stress resistance, any resistance. This slide is clear or not? Every participant? So these are some basic points about non-coding RNA. Now some basics about long non-coding RNA. Long non-coding RNA are RNA molecules that do not code for proteins but play a vital role in regulating gene expression. What are the characteristics? obviously length is more than 200 nucleotides. Lack of open reading frames, obviously that's why it is non-coding. Can be transcribed from intergenic region, enhancers or any other genomic areas. So these non-coding RNAs can be. Now there is a major difference that I have realized now between microRNA and lncRNA in their transcription. As we have seen in MIR base, they have given the gene IDs that are from which the microRNA are transcribed. It means microRNA can be transcribed from specific gene because they have given the IDs. Remember, in MIR base they have given the gene IDs, but for lncRNA there is no specific gene ID, no specific genomic location, no specific region of the genome. They can be transcribed from any region. Okay. Now is about biogenesis of long non-coding RNA in plants. They can be transcribed by RNA polymerase 2 or 3. They also undergo processing. They undergo capping, splicing and polyadenylation similar to messenger RNAs. So processing part is same. Stability, lncRNAs can be stable or rapidly degraded depending upon their functional role. Is this diagram clear? So this diagram is a well explained view of biogenesis of lncRNA, that how they form and depending upon the biogenesis how they are classified according to the different regions. So here they are formed in nucleus. This is the nucleosome depleted regions. Depleted region means from where the chromosomes are segregated. Yes. Now this is RNA polymerase 2. It is giving some premature transcripts. You have studied in transcription that it is not always that fully transcript is made. First some premature transcripts are released. These premature transcripts can be degraded and can undergo RNA processing. So they will form mature lncRNAs. How interesting is this? That some premature transcripts are even not waste. They are regulating the gene expression at so many different levels. So these long non-coding RNAs are formed from these premature transcripts. Have you understood this? Now these mature lncRNAs, they are classified according to the biogenesis, according to the region of the biogenesis. If they are formed from enhancer, then enhancer RNAs, promoter non-coding RNAs, then intronic region, intronic long non-coding RNAs, intergenic link RNAs, then that are formed by R loops, formation of R loops, and they are formed from negative strand of the genome, that is NADS. NADS is natural antisense transcripts because they are forming naturally in the organism. Antisense means they are forming from the other strand by formation of R loops. NADS are basically formed by formation of R loops. Is this clear? So these are the main classes of the long non-coding RNAs according to the formation, biogenesis from the specific regions of the genome. R loops are basically formed when the chromosome is depleted and the RNA polymerase is binded to the antisense strand. and that this loop formation is R loop formation. And these R loops are recently discovered, that during the formation of R loops, then only natural antisense transcripts are formed. So this is clear or not? Now is the functional role of lncRNA in plants. lncRNA can work at various levels: epigenetic level, transcriptional level, post-transcriptional level, and translational level. Epigenetic level is the epigenetic, means during the chromatin remodeling. Have you heard about chromatin remodeling complexes, PRC complexes, polycomb repressor? These proteins, these lncRNAs can bind to these complexes, PRC complexes, and from there they can regulate the expression of that genomic area at the chromosomal level, at the epigenetic level. क्लियर हो रहा है सबको कि बस ऊपर से कुछ नहीं आ रहा? Some basic points, very basic points. Gene expression regulation through chromatin remodeling and transcriptional silencing. Interaction with transcription, lncRNA can interact with transcription factors, promoters, enhancers. By interacting with the promoter of some gene, lncRNA can upregulate or downregulate the gene expression. There is no specificity that it can only upregulate or downregulate the gene. It can do anything to the gene, that depending upon the situation. Then is epigenetic level. lncRNA guide chromatin modifiers to specific loci affecting the DNA methylation and histone modification. During the opening of the chromatin, have you heard about euchromatin, heterochromatin? lncRNA can work at that level. Euchromatin is active and heterochromatin is inactive. Now there is something that convert the heterochromatin into euchromatin. There are various complexes, chromatin remodeling complexes, that convert that heterochromatin part to the euchromatin part, from which various genes are formed. So lncRNA can work at that level also, epigenetically. Then is post-transcriptional regulation, can interact with microRNA, as we have covered, competing for binding sites. Then is stress responses. lncRNA regulate responses to abiotic stresses, for example drought, heat, pathogen resistance, because it can regulate the expression of the genes that are particular to that stress. This is just the flow chart made by my colleague. So these lncRNAs, they can provide biotic stress tolerance. Some famous lncRNAs which are well characterized, well studied are Elena, Solanum lycopersicum LNC0195, then T-lncRNA5 for phosphate homeostasis. These are all plant lncRNAs, IPS1, OS, TPS, उसके बाद salt stress, DRIR, drought stress, auxin controlled plant development, vernalization, photomorphogenesis, nodule development, photosensitive male sterility. So various functions of lncRNA has been discovered. Further, these are the major lncRNAs, Coulier from Arabidopsis thaliana, that is involved in vernalization and flowering time regulation. Then rice, responsive to salt stress, regulating stress response genes. Then further immunity related lncRNA of Arabidopsis. There are various studies that has been published about lncRNA, but these studies are only about computational based. Very few studies of lncRNA functional characterization are published. So, functional characterization, these are the challenges of lncRNA research. Functional characterization, lack of clear and consistent functional annotation due to complexity of lncRNA functions. As lncRNA can do function at various levels. So, just computational prediction, computational pipeline generation, they can give some details of lncRNA, but actual function of lncRNA can only be characterized when they studies in planta, in vivo, in the lab, wet lab experiments. So, this is the lack of this research. Then techniques and tools, need for specialized tools. Like microRNA, we have studied various tools, well established tools, but for lncRNA, circular RNA, there are not that much tools that are available. For lncRNA detection, quantification and functional analysis. Species specific variations. Plant species may exhibit unique lncRNA profiles and functions. Like microRNA 156 we have covered, it has same sequence in grapes, in Arabidopsis, in rice, in soya bean, but lncRNAs, they have different sequences in all the species, but they have different sequences in the different tissues of the plant. Like grapes, I have small leaf, mature leaf and moderate leaf. These leaves can show different sequences of lncRNA. So, they are very specific to the state, to the time, spatio-temporal, we can say. They are very specific to the spatio-temporal part. So, this is also a lack part of the research of lncRNA. So, these are the major databases of lncRNA. First is plant long non-coding RNA database. This database consists of more than 13,000 long intergenic RNAs and associated epigenetic markers. Plant non-coding RNA database consists of 11 different type of non-coding RNAs of 150 plant species. Green non-coding RNA database consists of data of 37 plant species and algae with more than 1,20,000 lncRNAs. Next is TAIR, the Arabidopsis information resource. It can also have data of some lncRNAs of Arabidopsis. Then is ARAPOT. It consists of annotated link RNAs, NATs and various other long non-coding RNAs. Then is plant natural antisense transcripts database. It contains only NATs, natural antisense transcripts. It consists of NATs annotated data along with the expression of small RNA of 70 plant species. Then is Cantana database. It consists of data of 45,000 lncRNAs of 10 model plant species. So till here, have you all just go through the basic points of non-coding RNAs?